Okay, so hello guys, and welcome back to Agriculture Academy. So we were doing DNA repair. Okay, so in continuation with that, so now we are going to discuss about the replication errors and the repair or the nature of mutation and how these mutations or the replication errors which is happening is getting repaired or the rectified. Okay, so already we have discussed about the transitions and transversion. It's just the repetition is for you. So these are the main two basic type of mutations. Okay, so whenever we talk about like what kind of mutation is happening, okay, so basics ones are the transitions and transversion. Please remember, transition is like from pyrimidine to pyrimidine or purine to purine substitution. Okay, so instead of T, you are getting C. Now, when you represent any substitution mutation, okay, wherever if you're teaching or when suppose you are representing anywhere, okay, so instead of saying substitution word, what you can do is there is a terminology to use, and that is like T is getting substitution by C, okay, or A is getting substituted to G. So if you write like this, it means the person should be able to understand that this is substitution type of mutation. Okay. Okay. Of course, this will not tell that whether it's transition or transversion, but when you look at TC, we know that both of them are pyrimidine. So you will understand that these are transitions. Okay. Now, if you're having from T substitution to G, so I know this is pyrimidine, I know this is purine, but this symbol like this, okay, like greater than or a symbol, it represents that again it's a substitution type of mutation only. Okay, but transversion is happening. Okay, and all these mutations which is altering only a single nucleotide, they are known as the point mutations. Is this much clear? It's very simple, right? Just remember, whenever we write like this, it means we are talking about substitution. By looking at the pyrimidines and purines, you will understand whether it is transition or it is transversions. Clear? Yes, it is clear? Okay. Now, again, this is just the representation in the diagram way, in the diagram. So here again, transitions and transversions. So what are these? So A to G or T to C is your transition. Okay. Now, but if you have like A to C or C to A, T to G or G to T, all right, then again, G can go to C also, and C can also go to G, where A to T and T to A. So all these crisscross is the transversion. Okay. Now, can you calculate what is the probability of having transversions? If you have got A, T, G, and C. So how can you calculate the probability of occurring transversions? Yes. How will you calculate? So all these are happening in one position, right? So what is the chances that instead of A, you can have T or C? Out of 4, what are the chances of having T and C?
One by two.
Yes.
It is 2 by 4.
Yes. Either you say 2 by 4 or you can say 1 by 2, right? Now, since we are talking about one only, and the probability of occurring all these, right, is going to be 1 by 4. Getting the point? The occurring. Either it can be A, T, C, G, or C. So it is going to be 1 by 4 only in one position I'm talking about, right?
Yes, ma'am.
Yeah. It is not given that अगर क्वेश्चन में हमने बोला होता कि यहाँ पे A है, यहाँ पे G है, यहाँ पे C है. Now what is the probability of transversion occurring? तब आप इन 3 जगह के लिए कैलकुलेट करते हैं. Like in place of A, you can have C or you can have T, right? तो यहाँ पे हो जाता है 2 by 4. Now G में ट्रांसवर्शन होने के लिए again you can have either C or T, again 2/4, right? Now for C you are going to have either G or A, so again it is 2/4, correct? Getting?
Yes, ma'am.
So the transversion happening in this sequence is going to be 1/2, 1/2, 1/2, 1/8. तो हम ऐसे कैलकुलेट करेंगे. बट अगर मैं आपसे सिर्फ एक पोजीशन का पूछ रही हूँ, सिर्फ वन पोजीशन, सो व्हाट विल बी द प्रोबेबिलिटी देयर? फिर आप कैसे निकालोगे? Is this much clear?
Yes, ma'am.
ओके. अगर मुझे सिर्फ एक जगह का निकालना है, सो सिंपल, इट कैन बी एनीथिंग, राइट? सो हमारा आंसर 1/4 रहेगा. इट कैन बी एनीथिंग. ए हो सकता है, टी हो सकता है, जी हो सकता है, सी हो सकता है. हमने मेंशन नहीं किया. आई हैव नॉट गिवन यू द सीक्वेंस, राइट? अगर मैं आपसे ये बोलती दैट, अगर मैं आपसे ये बोलती कि यहाँ पे सिर्फ ए है, ओके, 1 सेकेंड. Okay, now if I say that in this position A is present, now what is the probability of transversion happening in this position? So आपका आंसर क्या होना चाहिए? So you can have C or you can have T out of 4, right? So this will be 2/4, 1/2. ठीक है? Now you got it?
Yes, ma'am.
Okay.
Yes, ma'am.
So these kinds of questions will come. Okay, आपसे डायरेक्टली ये कोई नहीं पूछेगा कि ट्रांजिशन क्या होता है, ट्रांसवर्शन क्या होता है. आपसे ये पूछा जाएगा कि क्या प्रोबेबिलिटी हो सकती है of mutation occurring. Okay, yeah. Now let's talk about like replication errors, which is escaping the proofreading mechanism. Now what proofreading mechanism? If you remember in the DNA polymerase, you had 3-5' exonuclease activity as well as 5-3' exonuclease activity, right? So ये आपका DNA pol 3 में है, ये 1 में है. So all this is required during the replication. Somehow all this mechanism के होने के बावजूद there are errors. Okay, now how to overcome those problems? Okay, so as we have seen, the replication machinery, it achieves a remarkably high degree of accuracy using proofreading mechanism. Okay, so we have got the 3-5' exonuclease component of the replosome, which removes strongly incorporated nucleotides. Now, proofreading improves the fidelity of DNA replication by a factor of 100. Now, when I say fidelity of DNA replication, what does it mean? Fidelity का मतलब क्या होता है? But however, it is not foolproof. Fidelity of DNA replication, fidelity मतलब क्या होता है? DNA replication के ऊपर आप कितना और भरोसा कर सकते हो, right? So that much 100 times it is improving, but it is not foolproof mechanism. कुछ ना कुछ error जो रहते हैं वो escape हो ही जाते हैं, ठीक है? Now, 3 different nucleotides can be misincorporated opposite to each of the 4 kinds of nucleotides, right? So what are the possible matches? Now अगर हम यहाँ पे ये देखें, so TGC opposite to T in one template. तो इसी तरह से अगर T के opposite में G आ सकता है, C आ सकता है, A आ सकता है. Similarly, G के opposite में T आ सकता है, C आ सकता है, A आ सकता है. So like this, total we are going to have 12 possible mismatches. Correct? Okay. Now, if the misincorporated nucleotide is not detected and replaced, this will become permanent in the genome. So we know what will happen then. As the replication rounds are increasing, the gene sequence will get changed. So obviously what will happen? Eventually, the gene change will have either a truncated RNA transcript or a protein, which is not going to be functional further in the cell. Okay, so this is going to be a permanent damage in the DNA sequence. अगर हमने इसको अभी correct नहीं किया तो, okay. Now there is one more thing that once you have all these mutations accumulated, the chances of having more mutations in the upcoming replication is also increasing, right? It's simple logic. जहाँ एक गलती हो गई है, the chances of, and if it is not corrected, so they understand that the chances of having more mutations in that particular area becomes more, right? They become more error prone, right? Okay, are you getting the concept over here?
Yes, ma'am.
Yes, ma'am.
Okay. Now, look over here. So replication change can change a misincorporated base into a permanent mutation. Okay, so you have got the first round of replication is there. Now let us see. Okay, there is a mistake happening. Okay, so A के opposite में T आना चाहिए, but क्या आया आपका G आया, ठीक है? So this is happening during the replication. तो ये आपका parental था. Okay. Now from the parental it is going. तो यहाँ पे एक strand में A के opposite में G आ गया. So this is your one mistake. But the other strand, other parental daughter strands, this is okay, normal, good. Now what is happening? Now second round of replication, this is going to give you A and T in both the daughter strands in the next round of replication. But here, it is going to give you AT and CG. So what had happened? This chromosome, sorry, out of 4, this chromosome is going to have a wrong incorporated nucleotide. यानी कि यहाँ पे आपका gene change हो गया है, ठीक है? And the problem is that if it is not rectified here itself, it is then going to become permanent. This is known as permanent DNA damage. Okay, I hope this is clear. Simple topic. Okay, now mismatch repair. So this is the first mechanism by which it removes the replication error. So let's talk about this mismatch repair. Important है guys, ठीक है? और questions, if you see, अगर आप previous इसके question paper देखोगे तो सबसे ज्यादा mismatch repair से ही पूछा जाता है. बाकी जो आपके base excision है, nucleotide excision है, homologous recombination method है, उसमें से इतना ज्यादा नहीं आता जितना ज्यादा mismatch repair से आता है, ठीक है? Okay, so let's talk about our mismatch repair. So here, look at the figure. Okay, मैंने थोड़ा short में लिया है. I have removed the description and kept the diagram more. So look at over here. So can you see there is a kink present, right, in the double-stranded DNA? Now what happens is that mute S, okay, it will go and start scanning the DNA. जैसे ही आपका DNA replication हो गया, okay, mute S will come and it will start scanning the DNA. Now once it is scanning, wherever this kink is present, जहाँ पे भी ऐसे bulge is present, what will happen? This gets detected by the mute S. Okay. Now, mute S, once it detects, is going to recruit your mute L and H. Can you see here? It is going to recruit your mute L and mute H. And the ATPase activity of mute S. See, can you see यहाँ पे आपका ATP binding domain है. Okay. Now, it gets activated and hydrolysis of ATP will happen. Now, once ATP is happening, what will happen? Mute H आपका activate हो जाता है and it is going to make a cut over here near to this side. Okay, can you see when mute L and mute H is recruited, ATPase activity gets activated of the mute S and mute H, which is a endonuclease, will make a nick in the strand. Okay, now what will happen? Exit nucleus will come. Okay, pi dash 3 dash exit nucleus will come. They will make a gap over here. They remove the जो wrong incorporated nucleotide है, it is going to remove. Then you know what will happen. RNA primer होगा, Okazaki fragments बनेंगे, ligase आएगा, and it will seal it. Correct? Okay. See. So exonuclease is there, then later your DNA polymerase 1 will come and then ligase will come and it will fill the gap. Okay. Now, any questions in this diagram or in this mismatch repair pathway?
No, ma'am.
Okay. So I'm going to ask you one question then. Yes. Anyone was asking question? It's clear, right? Should I repeat?
No, ma'am.
It's clear, ma'am.
Good. Now, I'm going to ask you a question. Mute S, mute H म्यूट L को कैसे पता चलता है इन दोनों में से कि T हटाना है या G हटाना है? हाउ डू दे अंडरस्टैंड? अभी तो हमने यहाँ पे ग्रे एंड रेड कलर दे रखा है, राइट? इवन वी डोंट नो पेरेंटल कौन सा है, डॉटर कौन सा है. सो हाउ विल दे अंडरस्टैंड दैट विच वन इज रॉन्गली इनकॉर्पोरेटेड, वेदर इट इज T और G? कैसे पता चलेगा?
Hydrogen bonds.
है नो, मिसमैचेस है, बट उसको कैसे पता चल रहा है कि मुझे डीएनए सीक्वेंस में T गलत आया है या G गलत आया है? Like, should I change this strand or should I change the bottom strand? उसको कैसे पता चलेगा? See, once DNA is replicated, sorry, okay, it is like this, ठीक है? Now inside the cell we don't have this color differentiation, right? कि parental ये कलर में है, daughter ये कलर में है. So म्यूट S, म्यूट H, ये जो सारे प्रोटीन्स है, रिपेयरिंग वाले प्रोटीन्स, उनको कैसे पता चलता है कि मुझे इस स्ट्रैंड से T हटाना है या ये स्ट्रैंड से G हटाना है? उसको कैसे पता चलेगा कि इन दोनों में से गलत कहाँ पे है, गलती कहाँ हुई है?
Is it the size? Because now, if it is T and G, both of the like one of them is supposed to have just one ring, the other is supposed to have two rings, so that will create a difference in the size on one of the sides.
No. So when these scanning is going on, so if म्यूट S, म्यूट H एक प्रोटीन नहीं होंगे ना. Suppose this is like, for example, I'm talking about the prokaryote के केस में. Okay, इतना बड़ा हमारे पास DNA क्रोमोसोम है बैक्टीरिया का. तो जैसे जैसे रेप्लिकेशन हुआ, म्यूट S and all these proteins will start coming in scanning. एक को यहाँ मिलेगा मिसमैच, एक को यहाँ मिलेगा. So it is not possible for them to scan the entire. तो 2-3 प्रोटीन आके वो स्कैन करेंगे, जहाँ पे भी मिस्टेक होगा they are going to rectify. But again, in that, how do they understand कि इसमें से नया कौन सा पुराना कौन सा है? I have actually given you the hint.
Ma'am, parent cells are methylated.
Yes, this is the correct answer I wanted to hear. Okay, yes. So how do they differentiate between parental and daughter strands? Is that parental strands are methylated and your newly synthesized daughter strands are not. So this condition is also known as hemimethylation, correct? We have discussed all this already. So when they scan
this happens right as replication is happening, right? Because almost immediately the daughter strand is also methylated.
Yes.
Won't be a difference after.
So before DAN methylase comes and methylates the daughter strands, this is also going on. See, कैसे है? Replication यहाँ पे ऐसे बढ़ रहा है, पीछे आपका स्कैन भी होता जा रहा है. अगर स्कैन होके आपको क्लियरेंस मिल गया है, then DAN methylase आके पीछे से it is methylating all your daughter strands also. See, हमें इतना टाइम लग रहा है पढ़ने और पढ़ाने में समझने में, but they are not waiting for us. Within a second, they are completing their job. Okay, so this is the correct answer, that they detect while scanning by checking the methylation group present. Okay, good. Okay, so I mean, I have asked a question before itself, but yeah, it's the same thing. So how does the E. coli mismatch repair system know which of the two mismatched nucleotides to replace? And yeah, so the answer is, it is the hemimethylation part. Okay, good. Now this question, so since I think I have told you I worked at NCL and then ICL Pune and then Tata, right? So of course at NCL Pune I was doing myself PhD. So but when I went to ICL Pune, so I was there as a postdoctoral fellow. So we used to sit for the interviews, like जो भी नए बच्चे आते थे recruit करने के लिए for PhD and integrated PhD. So there we used to ask such questions. So हम ऐसे out of the syllabus कुछ भी पूछते थे. So like I have said over here that replication हो रहा है, इतना ये हो रहा है. वहां पे सिर्फ हमने सिंपल डायरेक्टली ये पूछा था, हाउ डज द रिपेयरिंग एंजाइम्स, ओके, हाउ डज द रिपेयरिंग एंजाइम्स अंडरस्टैंड व्हिच स्ट्रैंड इज नीडेड टू बी रिपेयर्ड. सिंपल, हमने ये नहीं बोला है वो म्यूटस है, म्यूट एल है या एच है या प्रोकैरियोट्स है या यूकैरियोट्स है. सिंपल, हाउ डू दे अंडरस्टैंड हाउ द प्रोटीन्स आर एबल टू डिफरेंशिएट बिटवीन द पैरेंटल एंड द डॉटर स्ट्रैंड्स. And yeah, of course we get, अगर मैंने 100 बच्चों को पूछा तो 100 different answers we used to get except the right answer. Yeah. So yeah, so that's how I remember. So इसलिए मैं हमेशा बोलती थी, now also I'm saying the same thing, it is always good to study from the reference books, okay, as compared to the जो भी क्या कहते हैं academy का हो गया है या कहीं से भी आप पढ़ो. I mean, for your concepts clear it is good. जो भी आपके टीचर्स जहाँ से भी पढ़ाएं, it's good concept clear करने के लिए. But if you want to, you know, ऐसे interview अगर clear करना है in future, whether it is job interview or your PhD interview, कहीं का भी interview हो, ठीक है? If you study from these reference books, your basic understanding, your knowledge will be clear. तो आप कहीं का भी कैसा भी twisted question का आंसर दे सकते हो, ठीक है? So that's why I always prefer study from the reference books. उससे ज्यादा कुछ कोई नहीं पूछेगा, okay? Unless until that particular academy or institute is making a paper. तो आपका इंस्टीट्यूट अगर आपको एक्स्ट्रा पढ़ा रहा है तो वो क्वेश्चन डालेगा, अदरवाइज सीएसआर तो नहीं डालेगा, बिकॉज आई हैव ऑलरेडी मेंशन्ड दैट दीज आर दी प्रॉपर टेक्स्ट बुक्स और रेफरेंस बुक्स टू रेफर. ओके, whether you go to Pakistan or whether you go to US or anywhere, these are the reference books for these particular topics. Okay, so this is the same thing. This figure also I have shown before. So parental strand है आपका जो methylated है, and your newly replicated daughter strand जो है वो नहीं होते हैं. So this condition is the hemimethylated. Okay, so this is what we have like said. Now this is also functioning as like, you know, memory. Okay, so this acts like a memory. So mute H protein, they binds at hemimethylated sites. Okay, now when this mute H is contacted by L and S, which is near the mismatch, then only the H becomes activated, and your H is the endonuclease. Okay, so it selectively nicks the unmethylated strand, thus only a newly synthesized DNA in the vicinity of the mismatch is removed and replaced. So here the methylation is acting like a memory device of the this is in the case of the E. coli. यहाँ पे एक और क्वेश्चन मुझे आ रहा है. Okay, let's go to the figure. I'll just go back to the figure. Okay. Okay, so just see the figure carefully. अभी मुझे ये बताओ, how does the mute H, which is the endonuclease, will understand कि मुझे कट यहाँ मारना है या यहाँ मारना है? See, if I want to remove, अगर यहाँ पे G है तो मेरे को फिर ऐसे चॉप ऑफ करना होगा, राइट? तो नहीं इस डायरेक्शन में चॉप ऑफ करना है. अगर मैंने कट यहाँ मारा है तो मुझे चॉप ऑफ इस डायरेक्शन में करना है. So how does the enzyme understand कि मुझे किस तरफ जाके कट करना है? Question is clear? अक्षय, क्वेश्चन समझ में आया?
Yes, sir.
ओके, सोचो. Anyone? Any answer? Yes. श्रुति, क्या लगता है क्या होगा आंसर?
I'm not sure, ma'am. I feel like it might be mechanical because since mute S is traveling right after as the DNA is replicating, right? So it always has to be in one direction. It has to pass it before it manages to cut. But I'm not sure.
See, in the case of DNA, always remember one thing is something about the directionality. Okay. अगर कोई enzyme इस direction में जा रहा है, so what does it mean if I say like this? So what does the strand say? That this enzyme is going to be 5-3' specific endonuclease. अगर वो ऐसे जा रहा है तो क्या हुआ? 3'5' हुआ, right? So this is how you have got the protein. So अगर मैं ये कह रही हूँ ऐसे जाना है वैसे जाना है, it totally depends on the directionality. किस हिसाब से आपका DNA स्कैन हो रहा है and which protein will be recruited. ठीक है? I'll show you. So this is what is discussed over here. Okay, I'll show. So look it over here in the figure. So depending on that, you are going to have that particular type of endonuclease. Okay. Okay, so before the time gets over, look at the figure. So what do you see in the first figure? You have got the methylated, and lower one is your the daughter strand. Okay, so you can see how beautifully the mute H, L, and S is there. Mute S is scanned, has scanned the mutation or the mismatch which has had happened over here. Okay, फिर म्यूट L and H आता है जो ठीक कैन यू सी म्यूट L आपका जो रहता है वो मिथाइलेटेड में रहता है, उसके ठीक ऑपोजिट में H आता है जहां पे वो निक बनाता है. Now if it is 5' to 3', then it is in this direction. Can you see 5' to 3' exonucleus? It is also known as exo 7 or rec J. Okay, please remember, so they are in the 5' to 3' exonucleus or also known as exo 7 and rec J. So then what will happen? Then आपका polymerase आएगा, यहाँ पे वो bind करेगा, then lysase आएगा. Okay, now if it is in this direction, then you are going to have 3'5' exonuclease activity. Simple. Okay, so it is always about the directionality of the enzyme. कि मुझे किस जगह पे कौन सा रिक्वायरमेंट है, तो जैसा जो रिक्वायरमेंट होगा वैसा हम अपना एंजाइम लाएंगे. सो इट इज आल्सो नोन एज एक्सो 1. So you have got exo 7, exo 1. It is easy to remember like 5' 3' exonuclease activity, or it is 3' 5' exonuclease activity. Clear? Is it clear?
Yes.
Okay, simple. Just understand about the directionality over here. Okay. Now, in the case of eukaryotes, again, it is similar to the mismatch repair. We do have got the homologs. So instead of mutas, we have got the MSH, MLH, and PMS. Okay. Okay, so the indeed eukaryotes have multiple mute S-like proteins with different specificity, and one is specific for simple mismatches, whereas another recognizes small insertion deletions resulting from slippage during the DNA replication. What do you mean by slippage? अभी हमने क्या मार्क की थी? Just about the substitution, है ना? So this TG या TA जो भी आपका हो रहा था यहाँ पे, so it was all about substitution. Now, in the case of eukaryotes, we do have insertion, we do have deletions. What does this slippage means?
Is it something about DNA falling off prematurely or missing base pairs because of that?
Just be more precise.
Maybe it did not insert a nucleotide in its place, missed it. Kind of.
So, okay. So what happens over here is that because of the during replication, okay, sometimes instead of having strand like this, you might have strand like this. क्योंकि कभी-कभी सीक्वेंस में ही you have got the complementarity, right? तो जिसकी वजह से क्या होगा, यहाँ पे जो रहेगा वो यहाँ पे बेस पेयरिंग हो जाएगा या कुछ तो होगा, हेयर पिन लाइक स्ट्रक्चर हो जाता है. So when DNA polymerase is coming, it is not having the access to this. So वो क्या होगा, यहाँ से synthesize करेगा, यहाँ से synthesize करेगा. Okay. Later on, what will happen? This much particular sequence is not being replicated. So when this is stretched open, अब मैं इसको open कर रही हूँ, ठीक है? This portion. So whether it will come under insertion, etc. or deletion में आएगा. समझा? Hello. Understood?
Yes, ma'am.
Huh? So now the question is, अभी क्या होगा? See. So what will happen when this is opened? देखो जब आपने replication किया, अभी ये suppose आपका daughter strand है, ठीक है? So what will happen? This much region is not replicated, correct? So because of this, what will happen now? आपका parental strand इतना बड़ा होगा, मगर daughter strand सिर्फ इतना ही होगा. So that much particular area of the strand of it has been slipped. Okay. For daughter strand, now that particular region is being deleted. Are you able to get it? I don't have now proper figure in this. It's just a question came to my mind. But are you able to understand? Okay. Okay.
Yes, ma'am. But I have a question.
हाँ, पूछो.
Since DNA Paul depends so much on the structure of DNA, right? Directionality and reading everything. So how can it just miss this point? Because then the structure will be drastically different. How does it just skip past this thing that looks very different?
Yes. See, this is what when we talk about fidelity of DNA polymerase, we say वो ऐसा करते हो वैसा करता है. But the rate at which it is adding the nucleotide, right? It's like, see, ये आप समझो ना, उसके पास भी बहुत कम time है during which it has to replicate the entire chromosome. So even they have got the pressure of completing the entire thing. And when you do something in rush, there is always a mistake happening, right? So even though we know that helicase is there, जो सबसे पहले आपका strand open करता है, then SSBs आते हैं जो उसको single stranded रखता है, so that the DNA polymerase can come and do the replication, right? So these are the ideal cases, but you never know what might have happened. कौन से chemical agents was present during that replication. See, we are talking about prokaryotes, right? So I am going to add something to kill that particular bacteria. तो क्या पता उस particular agent के वजह से that folding of the DNA had happened during the replication, जिसकी वजह से mutation हुआ, और जिसकी वजह से next round of replication में the bacteria is not able to get replicated, not able to multiply, and that's how they are getting killed if they are not able to repair this thing. Okay, usually they can repair. Are you getting the entire scenario here?
Yes, ma'am. Yeah.
Okay. So if it is getting slipped, so slip मतलब किसी cases में वो insertion होगा, किसी में deletion होगा, ठीक है? It's not that I'm talking about कि वो बहुत बड़ा strand insert हो गया या ये हो गया. So this can be one or two base pair only. जैसे ये G है और यहाँ पे C है, ये आपस में base pair बना लिए. So it will be something like this, ऐसा. Not big, but a small, small bulging out like this. So G and C यहाँ पे base pairing कर ले रहे हैं, instead of यहाँ पे C और यहाँ G, यहाँ की जगह वो आपस में कर ले रहे हैं. So because of this, what will happen? There will be in the daughter strand the deletion of these two base pairs. Okay. DNA polymerase को नहीं समझेगा, वो आगे बढ़ जाएगा. Yes, we know that they do have got the exonuclease activity. What if it is missed? Then see, these are coming under the replication error, which escapes the proofreading mechanism of the DNA polymerase. Correct? So this is what is getting repaired with the help of the mismatch repair mechanism. जो mute S and mute H, mute L के बारे में भी हमने discuss किया. Okay?
Ma'am?
हाँ.
How does insertion come from slippage?
Okay, how? Okay, before, see, after insertion में कैसा होगा? In case already ये ऐसा था. Okay, this is like the parental strand was having this. Now the daughter strand was getting replicated, the strand got to open up. So when daughter strand is cutting, तो उसके लिए तो ये extra हो गया, which is getting replicated. So this much region got extra in the daughter strand. अब क्या होगा? हर replication में this is present. So this much region is inserted.
From where does it take, ma'am? If there is a bulge, so you said so the DNA poly will not recognize it, and there is a deletion. So
that is one scenario. That is one scenario. क्या पता, after the replication हो गया, ठीक है? This is done. Okay, यहाँ पे आपका deletion वाला आपको समझ में आ गया. So they have gone to the another cell. Now in this cell, some other agents might have resulted in changing the secondary structures of the DNA. Okay, so it has already all these kinds of such bulges are there in the chromosomal DNA of the bacteria. The next round of replication हो रहा है, ठीक है? So there, it is getting open up. जो extra bulges थे, which was not replicated. I was saying, see, it's just all imaginary situation. I mean, of course, the people must have found out in their research. Just for you and me to understand now, I'm just giving you imaginary situation. Okay. So deletion समझ में आया, ठीक है? Same way, just उल्टा सोचो ना. Like already जब आपका ये DNA replication होने से पहले, you had this extra bulge present. Okay. Okay, all these are present. Now, all this insertion deletion किस वजह से होता है? If you have got, you know, alkylating agents which is present in between of these bases, base pairing. Okay, for example, आपने if you have heard of ecridin or ETBR हो गया, so they all are responsible for this insertion deletion. या फिर वो insert करेंगे या फिर वो delete करेंगे. Now, insertion कैसे? अभी वो बीच में present है, so because of which what will happen? There are chances that एक सेकेंड ना मैं बनाने की कोशिश करती हूँ. Something like this, and in between, let us say that this is your ATBR or ecridin orange present. ठीक है? Now, what is happening? ये loop-like structure आ गया है. Okay? It is not accessible. अभी क्या होगा? First of all, this should undergo ऐसे repair mechanism. Now, repair कैसे होगा? First, you need to remove the ecridin orange. I don't know any enzymes which can remove it. So what they do is they will just cut this much portion and they remove that area. Okay, so वो कैसे करेंगे? This will be cut and remove. Now the thing is that this bulge is present. It is not accessible. Now bulge है वो ऐसे नहीं रहेगा. It will form a structure. If you remember in the RNA, they form like this. ऐसे they have got the base pairing with themselves. ये थोड़े जो होंगे, which is not possible for them to form the base pairs. Otherwise, rest all will be forming a hydrogen bond. Okay, then you will have like these structures. Yes. Okay, now this, just imagine a situation where all this is present and it is getting replicated. Okay, so आपका helicase, just imagine a helicase which was able to break this secondary structure and open this entire thing in the second round of replication. Now what will happen? This entire region is now opened. So for the daughter strand, this much portion is being inserted. ये आपके first parent में नहीं था. This is a part of your second. यहाँ पे हमने you know, we have added some agents to kill the bacteria, but somehow it survived. Now third round में for this it is going to get inserted. Okay, so because of which the insertion is again, you know, without any this region. Okay, this region, abhi imagine a situation where this much and this much gene is not going to match from the previous wala region. Okay, yes, from the parental, because of the insertion अगर हुआ नहीं तो इतना आपका भी replicate होगा. अभी third case में जहाँ पे ये open up हो गया है. Now because of this sequence is getting inserted now. Now when you open up again, this sequence is not going to match with its parental one. Okay, so अब क्या हुआ है? So from generation to generation, it is all getting changed. The sequence is getting changed. Now in the case of prokaryotes, we know there are no introns. So एक particular nucleotide भी change हुआ तो it's a huge change in its gene, right? So just imagine ऐसे बहुत सारे आपके eukaryotic chromosomes में होते होंगे. So we know that in the case of eukaryotes, there are hell lot of introns are there. So access तक नहीं पड़ता. It only affects when it is happening in the codon region, exon region, right? Okay, थोड़ा सा idea भी मिला now.
Yes.
Okay. So this we just discussed. So and they also hemimethylation समझेंगे and Okazaki fragments बनाएंगे. Finally, DNA ligase will come and seal the strand. Okay, so this is how your mismatch is done. And of course, in the case of eukaryotes, you need to remember about the pCNA also and all this thing. Okay, this is the same thing. कुछ ज़्यादा वो नहीं है. Now talking about the mutations which is caused by the base analogs and intercalating agents. अभी शायद थोड़ा और clear हो जाए. So once we are done with this repair and when you will imagine in your head, okay, कि हाँ ऐसे ऐसे होता है DNA के साथ. See, I have tried my best to get the videos also. जहाँ जहाँ पे requirement है. But in case if it is not there, then you guys have to imagine. Okay, and once the imagination is done, तब आपको ये easy पड़ जाएगा. So now let's talk about base analogs and intercalating agents. So what are base analogs? नाम से ही पता चल रहा है. So they are going to mimic the structure of the bases, ATGC. ठीक है? And intercalating agents means like in between the bases. So they hamper the base pairing between the base pairs. Okay. So mutations are also caused by the compounds like base analogs and the intercalating agents, which causes errors in the replications. Okay. Now, see, this is what I was telling. There is no as such as a mechanism by which the enzymes can pick the base analogs and throw it away. और इंटरकैलेटिंग एजेंट्स के बीच में ATBR आ गया, वो उसको निकाल के फेंक देगा. ऐसा कुछ है नहीं हमारे पास फिलहाल. Okay. So base analogs are structurally similar to the proper bases. It just they differ in the ways by which make them treacherous to the cell. It means what? Like deathful. Okay. Now, the base analogs are similar enough to the proper bases to get taken and incorporated during the DNA replication. See, even though we say that, if you remember in our previous first starting lectures, unless until आपका DNTP उसके बाइंडिंग पॉकेट में नहीं बैठता, उसके साथ इम्यूनो एसिड इंटरेक्शंस नहीं करता, उसके साथ जब तक Mg2+ का कोऑर्डिनेशंस इंटरेक्शन नहीं करता, तब तक it is not possible for any wrong nucleotide to be added. Correct? We have all spoken about this. Like this is how a particular or the ideal case of a DNA polymerase should add the DNTPs during the DNA replication, but somehow it is still making a mistake. Just because now here the nucleoside triphosphate, they are looking like the base analogs जो भी हम अभी add कर रहे हैं. They just look like proper bases or the DNTPs. Okay, because of which what will happen? Now they have got the base pairing mechanism different and it will lead in the mutations. Okay, I'll show you the example. For example, 5-bromouracil. Okay, look at the structure. So we can see it over here, 5-bromouracil. Okay, now look at the structure. So what is the mistake? So it's a uracil. Uracil किसके साथ base pair होना चाहिए? अगर उसको होना भी चाहिए, अगर like present in the DNA, it should be base pairing with A. U का A के साथ होना चाहिए. First of all, uracil shouldn't be placed in the DNA. Okay, just because it looks similar to your cytosine, it is getting base paired with the C. Okay, so जहाँ पे नहीं होना चाहिए, it is present over there. So this is one kind of mutation. Now talking about the ethidium, proflavin, and ecridin orange. So these all are your intercalating agents. ये क्या करते हैं? DNA के oh, I'm so bad at drawing. So what will happen? They will come in between over here. Okay, so यहाँ पे present हो जाता है. आपका ATBR हो गया, ecridin orange हो गया, proflavin हो गया. They come in over here. Now the problem with ecridin orange and ATBR is that, like, if you know, have you guys run a gel in your MSc practicals?
Yes, ma'am.
So which one you have used? You have used ATBR?
Yes, ma'am.
Okay. So how you are doing? Are you putting ATBR into the DNA sample or you put into the agar gel?
We put it into the
Gel.
agar gel.
Okay, so you put into the agar gel. So of course, I mean, it depends from lab to lab, it varies. So either you can put into the sample also, you can put into the agar gel also. So if you're putting into agar gel, so you know that जहाँ जहाँ पे आपका DNA होगा, so you know that अगर suppose आपका इतना ये gel है, so this entire region of the gel is containing ATBR. Correct? I hope you people are handling with the proper gloves.
Yes, ma'am.
Yes. See, you might have seen people working, bare hands with the gel and all. Like 2-3 बार तो ऐसा हुआ कि मैंने भी हाथ से ही directly पकड़ लिया, just because we were in so hurry and wearing gloves and all, discarding was taking time. Even we have done the same. So at a level it is okay. It's just that, see, why it is okay, because you know that our palm cells जो रहते हैं, every day they shed off the dead skin, correct? So that is good that at the next day it is getting shed off. But just imagine you have consumed ATBR, then it's a problem because वो आपके अच्छे cells के अंदर जाएगा. which is not going to get shed off the next day. अभी आपके हाथ में लगा है, so it's fine, it's okay. I'm not saying it's a good practice, but it's okay. एक दो बार चलता है, but not every time. There was this person who was working continuously with ETPR. वो ऐसे ही use करता था, like without gloves and all. So after an year, that person got the skin problem. Okay, and yeah, of course, it started like a tumor only, but yeah, so since we detected early, so it was cured properly then. I'm just saying that we need to be very careful. Even though हमें लगेगा कि आज मैंने TBI छू लिया है तो कुछ नहीं होता है, that's okay. एक दिन से कुछ नहीं होगा. It's always about the doses. It's always about the exposure, how much you are getting. Okay, anyways, we'll talk about agarose gel also in detail. Now, so the problem is क्या है? अभी 5-bromouracil है, so it should, instead of having with the base pairing with the T, it is with the, sorry, instead of having with A, it is having with the G. ठीक है? So now just remember 5-bromouracil, it is an analog of thymine. Okay, so the presence of the bromo substituent allows the base to mispair with guanine via a enol tautomer. So wherever you have got this double bond O, enol, keto tautomer हो जाता है वो आपका. ठीक है? So this is how it is able to make a 3 hydrogen bonds with guanine, and which is resulting in the mismatch in the DNA. Now again, intercalating agents, they are the flat molecules. Okay, so they contain several polycyclic ring structures, and they can get integrated into the DNA. So agents like intercalating agents like proclavin, ethridine, they all cause deletion or addition of base pair with a few bases. So इनकी वजह से क्या होगा? Additions and deletions हो जाता है. Now, when these additions and deletions are happening, it is going to change the translation of the mRNA, because the coding sequence, if you remember what is open reading frame, it is going to get shift, right? कभी-कभी क्या होगा? Because of this shift, the amino acid sequence is going to vary. Sometimes it is going to become a truncated sequence. Now, when you say truncated, what does it mean? It means that because of the missing of the few nucleotides, the codon gets changed because of the insertions and deletions, right? And instead of coding for certain amino acid, it will code for a stop codon, because of which the open reading frame stops in between. So it is going to give you a truncated polypeptide. Correct? So I hope you know how insertions and deletions is giving a truncated polypeptide. Is this part is clear?
Yes, ma'am.
Yes, ma'am.
Okay. Now, how does the intercalating agents cause insertions and deletions? So one possibility is again the slipping between the bases is a template strand. These mutations cause DNA polymerase to insert extra nucleotide opposite to the intercalated molecule. अभी क्या होगा? Just because it doesn't understand कि मुझे यहाँ पे क्या bases add करने चाहिए, so it is going to add few bases randomly. ठीक है? Now the question should come: why does DNA polymerase will do that? जब उसको सामने टेम्पलेट में कुछ नहीं दिख रहा है, why it is going to add extra nucleotide? ऐसा क्यों करेगा DNA polymerase? Tell me. सोचो. Yes, अच्छा क्या लगता है ऐसा क्यों करेगा DNA polymerase?
Because of the rate at which it is doing, it does not see. Maybe, I don't know.
No, even if I say that कि चलो ठीक है उसको इतना सारा काम करना है, it's under pressure, ठीक है, but जब उसको कुछ दिख ही नहीं रहा है, see the question is wrong हो जाता है वो समझ में आता है, like it's in hurry, okay? जब यहाँ पे टेम्पलेट में कुछ है ही नहीं, देख अगर A है तो T डालेगा, G है तो C डालेगा, जहाँ पे because of the insertions of these intercalating agents, it is not able to visualize what is there in the template. तो क्यों randomly nucleotide add करता है? I'll tell you. See, the answer is very simple. Instead of keeping a gap in the DNA strand, it is better to have a mutation. See, DNA polymerase do understand that I can't see the template, but it cannot keep a gap also. क्योंकि gap रहेगा तो क्या होगा? It is also giving a signal that if gap is there, it means something needs to be repaired, right? और अगर gap रहता है किसी भी DNA strand में, what will happen? It's like a breakage. तो क्या होगा? Entire DNA will be discarded. The entire cell is going to get die. तो DNA polymerase क्या सोचेगा? So it's better that I add certain nucleotides and later it will be repaired rather than dying now. हम यही सोचते हैं ना, मरने से अच्छा है कि चलो एक हाथ पैर टूट जाए, बाद में ठीक हो जाएगा. Are you getting the analogy? I mean, this is not the correct way to explain, but are you able to get the point?
Yes, ma'am.
हाँ, so मरने से अच्छा है कि चलो गिर जाते हैं और हाथ पैर टूट जाता है, because we know that आज नहीं तो कल हाथ पैर हमारे ठीक हो जाएंगे. Same analogy यहाँ पे है. So instead of getting killed, like instead of going for suicide, apoptosis, it's better that now I add random nucleotide and later the repair enzymes can come and repair the mistake. ठीक है? So with this thought, instead of keeping a gap in the DNA, it is always better to add random nucleotides. And when these are not repaired, okay, when these are not repaired, then it becomes permanent in the genome, which results in the mutations, which results in the gene sequence change. जिसके वजह से your transcription and translation is getting affected. Okay, is it clear the analogy which I tried to explain? Yes.
Yes.
Anything? Actually, you wanted to ask something?
No, ma'am.
Okay. Okay, guys, I'm stopping here now. Anything else do you want to ask?